Hi, everyone. My name is Jihang, Wang. So I was a dimensions fellow in research at the University of Iowa. So at a medical college, just like Yasmin just introduced. Okay, so I was working at the Strack lab. So it's run by a Dr. Stephen Strack, who is the principal investigator of the lab. So this is part of the Department of Pharmacology within the Carver College of Medicine in the University of Iowa. So as I said, Dr. Stephen Strack and also Dr. Ronald Merrill. So he was actually a research scientist in the lab, and he's actually mainly responsible for everything I was doing. Okay, so the background of my project. So I was working with mitochondria, which is actually you can think of as an engine for the human body. So it is like important for electron transport and ATP synthesis. So the only thing we need to know right now is so mitochondria can do fission and fusion just like cells, so they can divide or combine. So the fission is bad for your body, and fusion is good for your body. So that's all we need to know at this point. So during the fission process, so you can see they're just look at the picture, like the green and the blue proteins. So during the fission process, so the blue protein, which is located on the mitochondrial membrane, it actually can recruit the green proteins to the mitochondrial membrane, and then the green protein can split the mitochondrial membrane in half. So that's how so you can just know that the blue protein and the green protein are involved in the mitochondrial fission process. Okay, so and what we found before was that MFF, which is the blue protein, is one of the two proteins responsible for mitochondrial fission. If you mutate this protein, then it can change the fission and fusion process because it is responsible for the process. So that's why this works this way. Okay, so previously we have done some mutations on MFF. So these of the wild type at the 9EDs are just different mutations. And as you can see from those electron micrographs, so the mutation, on the right, the picture actually has a shorter mitochondrial length because it's more efficient. So my project on this part is just to find out the relationship between the neuronal survival and the mitochondrial length, because we're suspecting if you have a longer mitochondria, which is more fusion, then the cell should survive better under stress. So I did two assays to figure this out. So the first one was a luciferase assay. So luciferase is just an enzyme produced by cells that can produce light, basically. So I just measured the light produced by the luciferase. And actually, we found from this assay, so they largely agree with our expectation that the mitochondrial fusion, which corresponds to longer mitochondria, actually has a neuroprotective effect under stress. So those are just me doing a lot of pipetting in different wells, because each different well has a different transfection. Okay, so the second assay I did was a nuclear morphology assay. Basically, I just use a really fancy microscope, and then I just just look at each cell and see if they're alive or not, which is, these are two pretty representative pictures of the cells. So you can see this one, it's not really clear on the slide, but you can see this one is I have three channels that I used. It's a blue channel, the green channel, and the red channel, and this one is just overlap of all the channels together. You see this one, it has all the dendrites looks pretty healthy, and that one is all the dendrites were broken up, so which means that one is a dead cell, and this one is a living cell. So so other science is over. So that's just interesting pictures at this point. So this was just me doing that's a epifluorescent microscope that I was using in University of Iowa. So I was just trying to locate the cells, because if you find the cells, it's actually not hard to see if it's alive or dead. But the hard part is because the well is so big. So you have to locate where the cells are. So that's the hard part. So that's me transfecting the cells. So this is actually a sterile hood, because when you are actually working on cells, you don't want any contamination. So any foreign cells can fly in there and just give you a different stuff in there. So that's why everything is sterile. So this is me making gel for running Western blot. So you see on the picture on the right, so that's another student from University of Iowa, actually a summer student working at the lab, and Nandita. So this is me just running Western blot, which is basically you just hook up those electrics, so the current can make the proteins move at different speed. So those are all the fancy equipment, kind of like Lego, actually, when you were actually working on this, but yeah. So this was me working on radioactive stuff. So you can see, so this little instrument I was using was actually to detect the radiation before I actually start the experiment to make sure there's no radiation leaking out before I can start working on it. And you see like the vest I was wearing, like the blue one's actually pretty heavy. It's actually 20-some pounds because that's a special material, I forgot what it is, but it's used to protect you from radiation. And all the stuff like the buckets, like under there, those are radioactive waste. That's why there's like a special glass that's blocking it. Okay, so that's I did many presentations. Have many opportunities to do presentations. So like those are me in the group meeting presentations, and this was me in the poster presentation. So the right picture. So like the guy on the right is Wang, Dr. Ronald Merrill, who was primarily in charge of my project. Okay, so reflections and remarks. So what I learned from this internship is I reinforced like the biomedical techniques I learned at Cornell, which is very important. And also I learned some advanced techniques with instruments that might not be available at Cornell because University of Iowa is a bigger school, so it has like more money to purchase like those fancy instruments. So those are very important for me because I want to go to grad school, so those are the level of research I'm going to be doing in grad school. And also I had, just like I showed the pictures, I have many opportunities to present my poster and my lab and my oral day presentation also. And also, so this internship makes certain that I know that science is what I want to do for my life, so it's pretty important. And also, and last but not least, I built many good relationships with my mentors and coworkers, and actually I got one reference for my grad school application from Dr. Strack, so it helped me a lot. And finally, acknowledgements. So, of course, the Cornell Fellows Program and everyone in the CEC, and also the Dimensions Program. And also, I just want to mention those are the grants supporting our work, but those are by Dr. Strack. And also, Dr. Barbara Christie Poe, who cannot be here today, but she was my faculty sponsor, who helped me a lot. And also, Dr. Stefan Strack and Dr. Ronald Merrill from the University of Iowa. And also Dr. Craig Teague, who's my faculty advisor for my entire 4 years at Cornell, who helped me a lot on every level of advising. So, and that's it. Okay, thank you.